Hello and welcome. Today, we're diving into the fascinating world of environmental DNA, or eDNA. Specifically, we'll explore the computational journey that takes raw genetic material from water, soil, or even air, and turns it into valuable information about biodiversity. eDNA metabarcoding is a powerful tool, letting us see which species are present without needing to catch or see them directly. It works by sequencing short DNA barcodes from environmental samples. But getting from millions of sequence reads to a list of species requires a complex process known as a bioinformatics pipeline. Think of this pipeline as a digital assembly line for genetic data. First, raw sequencing data needs cleaning. We merge reads, sort them by sample, and trim away extra bits like adapters and primers. This is data pre-processing. Next comes quality filtering, where we discard low-quality or incorrectly sized sequences to ensure accuracy. Then, we process the sequences. Identical sequences are grouped together. That's dereplication, which saves a lot of computational effort. We also remove artificial sequences called chimeras, which can pop up during the lab process. A really key step is clustering, or denoising. Older methods group similar sequences into operational taxonomic units, or OTUs. Newer methods, using tools like Data2, aim for higher resolution, identifying exact amplicon sequence variants, or ASVs. Once we have these clean, representative sequences, OTUs or ASVs, we need to know what they are. That's taxonomic assignment. We compare them against large reference databases like GenBank or BioID, using tools like BLAST to assign names, species, genus, family, and so on. Finally, all this information is compiled into an abundance table, showing which taxa were found in which sample and how many reads support each one. This table is then ready for ecological analysis. Sounds straightforward, maybe? Well, analyzing eDNA data comes with significant computational hurdles. The sheer volume of data generated by sequencing is immense, often hundreds of gigabytes per run. Storing and processing this requires serious computing power, often high-performance clusters or cloud resources. There isn't one single perfect pipeline. Choosing the right software tools and tweaking parameters is crucial and depends heavily on the study design and the specific DNA marker used. Perhaps the biggest bottleneck is the reference database. If a species' DNA isn't in the database, or if the database contains errors, we can't accurately identify it. Many species, especially from less studied areas, are missing. Distinguishing real DNA from errors introduced during the process also remains a challenge, though denoising helps a lot. And comparing results between different studies can be tricky due to variations in lab and bioinformatics methods, but the field is moving fast. We're seeing better algorithms, more use of cloud computing making analysis more accessible, and user-friendly pipeline tools being developed. There is a big push to improve reference databases and standardize protocols. Machine learning is also starting to play a role. Bioinformatics pipelines are absolutely essential for unlocking the potential of eDNA metabarcoding. Despite the challenges, continuous improvements are making eDNA analysis faster, more accurate, and more accessible, cementing its role as a game changer for understanding and monitoring life on our planet. Thanks for tuning in.